Getting started with our conservation webinar, I'm Holly Kirkendall, National Technology Specialist for the East National Technology Support Center. I'm pleased to turn the webinar over to Bill Holman. Dr. Holman is a wildlife biologist on the National Wildlife Team, and he's located in Fort Worth, Texas. Bill, you may now begin.
Thank you, Holly. Let me add my welcome to participants in today's NRCS Conservation Webinar. Thank you for your participation. It's my pleasure to introduce today's speaker, Dr. Christian Krupke. Dr. Krupke is a professor of entomology at Purdue University with both research and extension responsibilities. His work focuses on applied questions in the production of field crops, primarily corn and soybeans. A recent research focus has been quantifying the costs and benefits of neonicotinoid insecticide use on corn and soybean production. Despite the fact that virtually all corn and a vast majority of soybeans are being treated with these compounds, there is surprisingly little information on the efficacy and longevity of these compounds in target plants. Dr. Krupke's research couples the analysis of production costs and benefits with measurements of pesticide residues during and after planting in non-target plants, land, and waterways in order to allow for an assessment of the degree of risk to sensitive non-target organisms, including pollinators and aquatic organisms. Welcome, Christian. The floor is yours.
Thank you, Bill, and hello, everyone. What we're going to talk about today is run through a series of experiments, most of them performed by my lab group and collaborators here at Purdue University. We're going to look at efficacy, non-target effects, and best practices of neonicotinoid insecticides. Now, up front, I'd like to acknowledge many collaborators on this work, both students, postdocs, and researchers at Purdue and other universities. Most of this work is very collaborative in nature and wasn't just done here and wasn't just done in my lab group. I'd also like to acknowledge our funding sources, which include grower-funded organizations, checkoff funds, and the USDA. Now, there's been a focus on neonicotinoids for a number of reasons. The pollinator issue, which I'll be talking a little bit about, has taken the forefront. But the focus on these is justified in terms of their use rates. Most of our large acreage crops are grown from treated seeds. Clothianidin or thiamethoxam are the two neonicotinoids used there, and those are the two that I'll be talking about today. Virtually all corn contains one or both of these compounds, and 75% of soybeans are treated, and that's usually with thiamethoxam. There are other crops that are treated as well, as you see here, and there's been rapid registration, rapid adoption in the last 10 to 14 years. So in all, we use about 150 to 200 million acres annually of these neonicotinoids as seed treatments. They are derivatives of nicotine, as the name would suggest. They're neurotoxins that act on the central nervous system of insects, and they're effective against many insects, especially the sucking insects like aphids and whiteflies and some soil pests. Now, their adoption has been driven by a couple of key factors. The low vertebrate toxicity is first. High water solubility is also a factor, and I'll talk a lot more about that later on. But what that means is that there's systemic action in plant tissues. The plant can take up the insecticide with water in the xylem, so it moves up from the roots and into the plant tissues, and that can offer some protection to plant tissues that contain the insecticide. Now, these products are very toxic to honeybees and other pollinators, and you can see there just a few billionths of a gram are enough to cause mortality in honeybees. Now, what that means, the purpose of that number for now is just to inform everyone that when we use these products, everything has to go right. Everything has to go very well. The margin of error is small when you're dealing with compounds this toxic and on these large acreages. Now, these are a couple of graphs that I'll show you from the USDA that show the rates of clothianidin and thiamethoxam use both in 2003. Now, this is the year before the conditional registration for clothianidin came into place, and you can see nothing happening on the left image for clothianidin, a little bit of thiamethoxam use in some patches, as you see there. Now, below each map, you'll see the the use by year and by crop as well. And the gold bars, this is a little bit of a, we'll sort of lead you up to the next slide, it, represents use in corn, which clothianidin is by far the major player there, almost the only player. And the green is soybeans, red is cotton, and so on. Now let's fast forward to something closer to current. 2014, you see the use rate in those 11 years between these two images has increased dramatically, especially in the areas where corn and soybean are grown most intensively. Where I am speaking to you from, Indiana, is right in the heart of the region that is the most, that has the highest levels of use. Now, interestingly, in the last 5 years, we've had a bit of a spike in use in corn. This is data from some collaborators of mine at Penn State University, Maggie Douglas and John Toker. They published this initial image in 2015 in Environmental Science and Technology. Now, follow-up work, since that paper was published, since that analysis was done, resulted in this image in the bottom right. And you can see that what's changed here is that the use in corn, if you look at the scale on the left and look at the size of the corn, the magnitude of use in corn, which is the red or maroon color, has increased dramatically in those 5 years. Now, this isn't based on any increase in pest pressure or a new pest or damage or anything like that. But nevertheless, these rates have doubled in the last 5 years. So while that figure that I showed you a moment ago, that map of the United States, showed a dramatic increase, if we look at 2015, 16, and soon 17, we'll see even more increases, simply because there's more on every single kernel of corn than there has been in the past. Now, the first work that we did involving neonicotinoids began about 5 or 7 years ago, and was generated by a report of dead bees intersecting with corn planting in time. And dead bees outside of a beehive, if you see large numbers of them, always indicates that something is wrong. Of course, bees die all the time, but when you see a lot of dead bees outside a beehive, something is amiss if the bees can't keep up with the mortality and move the dead ones away. And what we found there is that there's an intersection between corn planting and when bees are moving around, and you see that image here. When bees are increasing in numbers is the blue line. You can see there's a steep increase as we get into the spring months. That tracks relatively well with when corn planting happens. And this is Indiana corn planting, and it spans from the middle of April into early June. And of course, the width of that green would move back and forth depending on the year. This year is going to be a bit of a later year due to the rain and so forth. But that intersects with when bees are flying and moving and building up their numbers. They're foraging actively. There are a lot of spring flowers. That's pollen and nectar sources that the bees need to survive, not just honeybees, but all bees. Now, bees, as they fly through the air, they become charged, similar to the way that we would build up a static charge if we shuffle across a carpet. Usually that's an adaptive thing. That's a positive for the bees because they can land on a flower, and any pollen on that flower will then adhere to their bodies, and they're passively collecting pollen, which they can later scoop off and groom off their bodies and put into their pollen baskets. You see a bee with a loaded pollen basket here, the hind legs. Now, when they fly through clouds of dust that may have neonicotinoids in them, then they pick up those compounds very readily because of this same static charge. And that's why about 5 or 7 years ago when we first began this work, we found that these dead bees that we found outside of hives coinciding with corn planting nearby all contained lethal levels of neonicotinoids on their bodies. There were also high levels of neonicotinoids in the hive itself. So what that led us to is to look at how is this happening, how far, what is the magnitude of this occurrence, what is the magnitude of the spread of these compounds. Now, we know planter dust is highly toxic to bees. That's some work that's a little bit older now. I'm not going to talk much about that, but most planters that we use to plant corn seeds and soybean seeds are what we call pneumatic or air planters. So they move a lot of air through the system to move seeds around, to singulate seeds on the planting disc, and to make sure that they drop at the chosen interval in the row. Now, when you're moving a lot of air through the planter, that air has to be exhausted as well. It's exhausted up and away from the planter, it's exhausted behind, it's exhausted down, depending on the planter manufacturer and the model. But in all cases, some sort of a seed lubricant has to be used. That's typically a talc graphite mixture that helps the seeds remain smooth and reduces friction between them and makes sure that they don't clump together so you don't get doubles or skips during planting. That exhaust dust, the exhaust talc and graphite that's been in contact with treated seeds, has very high levels of neonicotinoids in it. So the purpose of this next set of experiments is to look at how far that dust moves. And you can see here we have a schematic of a field. We have transects surrounding the field in all 4 directions. I only have 3 included here, but we do go to the north as well. And then we set out these dust collection stations at 0, 10, 50, and 100 meters beyond the field. And what these consist of is a vertical and a horizontal collector. The vertical points toward the field. The horizontal is at a height of about 30 centimeters above the ground. And that's to approximate a flowering plant, let's say dandelions, which are common around agricultural fields at this time of year. Each of these dust collection stations, you see the close-up there, is a series of 5 microscope slides. They're treated with a sticky coating just before planting begins. And then they catch any particles of dust that are moving both outbound in the vertical and particles that are landing on the horizontal slide. And you can see how we assemble those here. Once we have those slides, we take them back to the lab, wash the slides in solvent, and conduct LC mass spec analysis to determine not just which pesticides are there, but the quantities of them. This is a very sensitive way to detect large numbers of pesticides and to get to a very, very high level of resolution. So you can get down to 0.1 parts per billion. And we do need to go to a low level of resolution because, as I said earlier, very tiny quantities of pesticide are sufficient to cause mortality in honeybees. Now, this just shows what we saw when we were planting these. You can see this on the bottom right here. We're just putting some of the sticky coating onto the slides right before the planter moves through the field. This is a quick summary of how many slides we looked at. 6 slides per collector, 4 distances, etc. Over 1,000 slides analyzed for these data. Now, what we're after here is to look at the concentrations both adjacent to the fields, where we know, of course, they'll be very high, and people don't usually put honeybee hives right beside the field. But we wanted to know the beyond part of it. So the shape of that shadow, that kind of purple shadow that moves beyond the field, what is the shape of it? What is the magnitude of the concentrations as you move out from the field? Now, bees, of course, forage. And in the case of honeybees, they forage quite a long distance. They can forage up to a mile, typically. They can forage further than that if they need to. But in most cases, a mile or so is a fair estimate of a median foraging range. And that's denoted by that pale blue circle around the beehives there. That's the area that the bees forage, so that's the area we're interested in. Looking just at the beehive itself and seeing if there's deposition there is a first step. But since bees forage so widely, we have to consider how far they move, as well as how far this cloud moves. Now, this is the horizontal slide data. Each of these circles is a data point, and you can see most of our deposition is downwind. You can see we're still getting, in terms of nanograms per slide, we're still getting relatively high concentrations, even as we get out to 100. So what this tells us, we didn't go out far enough. We could have gone out farther. We do get some upwind deposition as well. It's not solely downwind. On the vertical dosimeters, again, we still have detections up to around 30 or so nanograms per slide downwind. And so again, didn't go out far enough. All the analyses that I'm going to talk about here only go as far as our data, so we don't extrapolate. You can imagine we could draw a curve beyond the 100 and take it out further. We didn't do that. We'll stay within the confines of what we recorded. So now we want to take these data and develop a risk map. And again, these are the components we're looking at. Now, we have data available that we didn't have to collect. The hive locations and crop type data, land cover, Those are all publicly accessible and available within different website resources. Wind velocity we collected at the time that we did the experiments, that we planted those fields. So we have all of these data at the respective locations here, FieldWatch, the USDA, and the USGS. And what we did with that is we basically began by mapping all of the Indiana cornfields, and you can see that on the far left slide. That's just the Indiana cornfield colored in. 2.2 micrograms per square meter is our deposition right there. That doesn't mean a lot now, but I'll hopefully make it relevant in a moment. Then we go 30 meters beyond, and we see what our deposition was, 30 to 60 and 60 to 90. Now, based on these analyses, 23.8% of Indiana apiaries get direct deposition, and the rates of that are 100 times below the contact LD50. So that was good news. So they're not, these apiaries are not expected to directly receive levels that would cause acute toxicity, that would cause mortality. However, foraging bees move. They move quite a distance across the landscape. And in fact, the bees that we found years ago that were killed were foragers in most cases. So there was evidence that they were picking these materials up, these neonicotinoids up, away from the field. So we assume a 1.4 kilometer foraging radius, that's approximately 1 mile, and these are blue dots on Indiana here are apiaries that we took from the FieldWatch website. We then drew a 1.4 kilometer radius around each of those. And we assume no static charge here. I talked a little bit about the static effect, but we omitted that here. We wanted to be very conservative in this analysis. So we just took the bee surface area, which is just over a centimeter squared, as shown here, and we take that surface area and move these bees through these dust clouds as they're there during the day. And we, again, assume no drift beyond 100 meters. Now you can envision what we did here. Here's a cornfield, here's an apiary, and we move this bee at random to forage in areas 1.4 kilometers from their apiary. Now what we found here is that the vast majority of Indiana honeybees will encounter dust during planting, depending on how many flowers they stop at, and we did a range of simulations there, and where those flowers are, we can exceed lethal doses in this case. Again, this is a conservative analysis because we only went to 90 meters. We omitted the static charge. We didn't include any other neonicotinoid-treated crops. So we didn't include soybeans in this analysis, even though they are, the majority of them are treated as well. So with this analysis, including the foraging component, less than 2% of Indiana apiaries are outside the deposition zone, again, assuming no drift beyond 100 meters. So the notion of moving bees to a safe area during corn planting is not realistic. There is no area where we could say they are quote-unquote safe. So whatever the solutions may be, that's not going to be a realistic one. I'll talk more about solutions toward the end. Now, what about benefits? As you saw, these materials are used on many acres, many crops every year. They're labeled for control of many insects. Most of them are below-ground pests, and you can see a sampling of them here. There are many more than these on the label, depending on geography, but these are the major ones through most of the upper Midwest that would be under consideration. We looked at efficacy of these products in conjunction with the dust study that I just mentioned to you. We looked in 3 areas in Indiana, 3 of our research farms. And these are areas where we typically look for efficacy work. We try to plant trap crops. We try to do minimal insecticide use here because we're trying to get pest infestations. For those of you not familiar, this part of Indiana, basically the northern half or northern third, is where the most intensive agriculture happens, and certainly the vast majority of corn production happens here. You can see our treatments here. We have naked seed. We have seed treated with the low rate of clothianidin and seed treated with the high rate of clothianidin. The data we're collecting here are stand counts, plant heights, root injury, and yield. And this is typically what we will do when we do an efficacy study in corn. I'm not going to show you all of the data. I'm just going to focus on yield. That's the one that most people are interested in, certainly most growers are interested in. And we didn't have any significant benefits in terms of yield from naked seed, versus the treated seed, Poncho 250 and 1250 in 2012, and similarly in 2013. Now, I'll talk a little bit more about why we saw those results in a moment. Now, what happens to the neonicotinoids applied to corn seeds? We've seen these results before where we have these inconsistent or a lack of benefits. Benefits are hard to quantify in corn seed, and looking through the peer-reviewed literature reflects that. The rates that we're using them and the acreage that is devoted to their use is not borne out by peer-reviewed publications showing benefits over a wide area. So one of the hypotheses we had is that neonicotinoids aren't in the plant long enough to provide benefit. So this is a schematic of a corn seed that's treated with a neonicotinoid. 1 to 2% is lost at planting. I talked to you about that, the abrasion. How much is taken up by the plant? If we can get that number or get an idea of what it might be, we may be able to assess what our pest management window might be. How long are these plants toxic? Now, we did this work at one of our research farms again to define what we call here the pest management window. This is a commercial hybrid, a DeKalb hybrid. You can see our treatments here. The first two have stars beside them because they're not commercially available to growers. Growers cannot buy naked seed in the commercial, in the elite germplasm they want, nor can they buy a fungicide-only seed at present. The bottom two are more commonly what you see, although the low rate of poncho is becoming less common. The high rate, which is also called the rootworm rate by most producers, is 1.25 milligrams of clothianidin per seed plus fungicide. And that's the subset of data that I'm going to show you. It's the most interesting. What we collected here are 10 plants were removed and photographed from each of these plots, each of these treatments per week, 3 times a week. The presence of pests and damage was documented, and the root, shoot, and seed were cut up from each plant and then homogenized. And the neonicotinoid content measured, again by LC mass spec as in the previous set of experiments. Now again, this is the 1.25 milligrams clothianidin per kernel. This is 6 days after planting. And this is what a plant or a seedling looks like 6 days after planting. It's not above ground yet. And these numbers are not going to mean much right now, but what they do show is that there's material moving from the seed, which is the 44 parts per million, and that material is both in the root and up into the shoot and the developing foliage of the plant. So all the data I'm going to show you are in this similar mode. We don't have any LD50 data for what concentration here would kill a rootworm or a wireworm or anything like that. So the approach we're going to take is to say when the concentrations approach 0 in the plant, then we can safely assume we're not getting any pest management benefit. In other words, when it's no different from a naked seed, well then our window of control is over. Okay, now what we saw was these concentrations follow an exponential decay curve, which just means they decline relatively rapidly. That's a schematic there on the left and the actual data on the right. And you can see in the seed we have the highest concentrations, as you would expect, but even then, by the time we get to 17 or 18 days after planting, we're approaching 0. And we approach 0, and we never get those higher concentrations in the shoot at the high rate of Poncho 1250 or 1.25 milligrams per kernel, or the root. Now, what do those neonicotinoid levels mean for a pest? Well, we chose rootworms because that's our key pest. This is also called the rootworm rate by producers. And this is what a plant would look like at 28 days after planting. Now, you look at the data I just showed you. You can look in more detail if you want to see the paper. It's freely available online. At the bottom right there, you see the information. But at 28 days after planting, there's not material left in the plant. So expecting the foliage concentrations, or in this case the root concentrations, to deter pet feeding is unreasonable. From 1982 to 2014, I show here the dates when the rootworms emerge, and it roughly aligns with 28 days after planting. So from that, we infer that the neonicotinoid levels, the fit with the key pest, in this case the rootworm, is poor. There are other pests, as I mentioned earlier. There are wireworms, white grubs, and so on, but those are relatively inconsistent, uncommon, what we call secondary pests. They're not likely to be present in many fields at economic injury levels, whereas the rootworm is an annual pest, or rather a perennial pest, the key pest of corn across most of the corn belt. So in this same study, you'll recall that we dug up the plants, looked for seed and seedling feeders, looked for damage, wanted to see some of these sorts of images. However, we didn't see any of that in this study. And that reflects that these pests, as I said, are uncommon. There's been a hypothesis put forward that in 2003 and 4, when these pesticides were registered for use in corn, that they were more common, and that we've now effectively removed them from the soil. And that may be true, that we have no data on either side of that. We don't have any survey data going back, 10 or 15 years to see if these populations of these secondary pests have gone up or down or stayed the same. But certainly in the recent work that we've done and that others have done, we don't see the pest populations that justify using these approaches to manage the pests that are on the label. Now, we filled in another section here of our schematic corn seed. We look at how much is taken up by the plant, and in that work that I showed you earlier, that's my Ph.D. student Adam Alford's work, we show less than 2% is taken up by the plant. So it's an inefficient, although there is systemic activity, it's relatively an inefficient way to get the material into the plant. So where does the rest go? So we still have over 90% left to account for. And as you can imagine, with very high water solubility, which the neonicotinoids have, particularly thiamethoxam, you do get movement. There was work led out of Jason Hooverman's lab in forestry and natural resources here at Purdue. I'm not going to talk too much about this. Again, this is freely available online, open access publication, and this was a field and a mesocosm study in Indiana. Findings I list here. We found neonicotinoids virtually everywhere we looked. This has been found in other Midwestern states. That's not a new finding. We did find some very high detections, particularly of thiamethoxam. That's well over levels that would be expected to cause acute mortality in many organisms, and developed some LC50s for resident aquatic invertebrates in the area. Daphnia magna is on the in the application package for neonicotinoids. It happens to be very durable in terms of neonicotinoid susceptibility. They're not very susceptible. They're difficult to kill. The aquatic invertebrates we looked at, including giant water bugs and damselflies, back swimmers, are far less tolerant. We also observed some sublethal effects on the predators in that they ceased feeding following exposure to sublethal levels of neonicotinoids. There's a lot more on this topic in another similar webinar by Christy Morrissey at the University of Saskatchewan, so I would urge you to check that out. She delves into this in much greater detail in her talk. Now, our other main crop in the upper Midwest and our nationally as well are soybeans. And soybeans, as I mentioned, the vast majority are treated with neonicotinoids. There is still some availability of neonicotinoid-free seed, unlike in corn. So we wanted to examine the utility of those neonicotinoid seed treatments for our key pest, the soybean aphid. Now, we have a couple of approaches for managing this pest. We have foliar sprays at threshold. There's a threshold that's been developed over 10 years ago of 250 aphids per plant. That's an action threshold. When the aphids exceed that number, producers are urged to treat them with foliar insecticides. Those are usually pyrethroids. There are a wide range of them available. Neonicotinoid seed treatments are also marketed toward aphid control and control of other soybean pests. And what we wanted to do was compare these two using a field study in multiple states, multiple years, to see where is the efficacy, what are the benefits, again, quantifying the pest management window as well. Now, for this pest management window component, we have two treatments, a fungicide-only treatment and a fungicide plus thiamethoxam insecticide. And you can see I have the trade names of those treatments there in brackets. So for this part of the study, we're collecting the leaf tissue. We're looking above ground because soybean aphids feed above ground, so we're not doing the root work here. We collected the newest unifoliates and trifoliates throughout the season, and we used an LC tandem mass spec again to compare the levels of thiamethoxam in those tissues. And these are the results. You can see the growth stage at the far left, days post-planting, and then you can see our concentrations. And you can see at the beginning of the year, just right when the plant emerges, and also in the cotyledon stage, which is VC, we have very high levels of thiamethoxam in those tissues. And again, when we look at the VC stage, we're looking at the cotyledon. So we're looking at basically the components of the seed. So it's not surprising we have that 9,000 part per billion there. However, again, that drops off relatively quickly, similar to the corn story. So what I've covered here is up to V2 when a plant looks like this. And that's our last significant difference between a treated and untreated plant, so 18 days. So whether we get pest management with 151 parts per billion on a V2 plant, I don't know. I would suspect not, but we've not looked. The larger point here, though, is that from V2, so from 2 trifoliates onward, we have no significant differences between untreated plants and the plants treated with thiamethoxam. So putting that back into perspective of what the soybean aphid, our main pest, their biology, typical aphid arrival is R2 or R3, and you can see this table doesn't even go that far, but certainly by that time you're long past the time, you're weeks past the time when you would have significant levels of thiamethoxam in the tissues. So not a good fit there. And we took those data, and those are that's sort of the mechanism behind how these products work. We wanted to look at, in a field situation, what are the break-even probabilities? Every year growers plant soybeans. Every year soybean aphids are a potential threat in some of these regions. In some areas they're more common than others, but in any case they need to be accounted for. There needs to be some assessment, some appreciation that this is potentially a pest that could impact yield. So we did a 2-year, 7-state field study here, and again this is in the upper Midwest. In 2012, none of the locations were over the soybean aphid threshold. The IPM option had a higher break-even probability, and the IPM costs there are primarily scouting, whereas on the cruiser treated seed side, we pay for the cost of the seed treatment, of course. In 2013, we had aphids over threshold in some locations, and the IPM option, as you would expect, did much better. We had over 98% chance of breaking even or better, whereas in the cruiser case we had about a 60% chance. And this paper is available online as well. You can see the link at the bottom right, or bottom left rather. Not surprising that an IPM approach where you're actually looking for the pest before you treat, responding to something that is over threshold and is known to cause damage at that level, would do better than an approach where it's not responsive at all. It's a prophylactic approach, so by definition, it doesn't react to populations of pests if they don't show up, or if they show up when the seed treatment isn't present, which our data indicate is often the case, then you can't realize a benefit from that approach. Now, I'm going to switch gears again and go back to the honeybee story a little bit. One of the areas we wanted to look at is what is the contamination of pollen through the season. So assuming we could nip this dust problem in the bud in terms of neonicotinoids, we know they're systemic. I showed some data that shows that they get into the water quite readily. Others have shown that in other states as well. What we wanted to ask is, how much of this material gets into pollen? And to do that, we set up honeybee hives adjacent to treated and untreated cornfields. We fitted them with pollen traps, which is shown here at the left. And every week, we would pull out the drawer of that trap and collect any of the pollen. You can see the pollen is sorted there in the lab down at the right corner. And again, this work is also freely available, open access at the bottom left. You see the link. So we had 3 sites here. We had a natural area, which is sort of an idyllic kind of meadow scene. We have an untreated cornfield, and we have a treated cornfield. Now, this is the course survey of the pesticides we found. We didn't just look for neonicotinoids. We're looking for all pesticides. We sampled for a total of 67 pesticides, both homeowner products, agricultural products, everything we might expect to find. The bottom line from this slide is that we find a lot of pesticides. We never found 1 or 2 or 3 per sample. We always found many. So one of the take-home messages here is that although we have to look at individual compounds when we do a lot of this work, in the lab, for example, working with honeybees, they do not encounter that in nature. They encounter blends and mixes of pesticides, and so it's important to bear that in mind. Those experiments are much, much harder to do, but people are starting to work in that area, realizing that combinations are different and will have sometimes more than additive toxicity. Sometimes there'll be synergy there. Now, this is the non-agricultural area in terms of pollen species. We're not a pollen species yet. We're still looking at insecticides, or pesticides rather. Look at the scale, the vertical scale. We go up to 350 ppb. This is a very coarse view. All we're looking at here is the additive parts per billion of all the pesticides. In the paper, we incorporated a toxicity value. In other words, if you have just a very tiny amount of a very toxic pesticide, that's more important than a large amount of a pesticide that's relatively non-toxic to bees, let's say. So we get more into that analysis in the paper, but here I'm just giving you a coarse view just to show you this, the gray and the purple bars were the key players here. Those are both insecticides. They're both pyrethroids. Perlethrin is the gray and the purple is phenolphthrin. These are not agricultural pesticides. These are pesticides that are used primarily in municipalities by homeowners and so on for control of nuisance pests. Those include things like hornets and mosquitoes and so forth. And if you look at some of the labels at your home or garden center, you'll see these active ingredients are there. They're not used agriculturally. They're not used by corn or soybean producers at all. Don't have a label. This is the untreated corn. The first thing I will show you here is that the scale changes. We're up to twice as much, so we're getting more material. But again, that's driven primarily by a non-ag compound. And again, there's the phenolphthrin here late in the season. Neonicotinoid-treated corn, and now the scale doubles once again. So we're getting more. Again, phenolphthrin is primarily the driver here. We have other pesticides coming and going. The neonicotinoids, many of you are probably wondering, they were a player here. You can see clothianidin is the blue, and that's shown in the 20th and 27th of June. You see a little bit of that. When we did the analysis, including toxicity, the neonicotinoids ranked in a high tier, although not as high as the pyrethroids did. So the pyrethroids, by virtue of how much was there, were the key players here in terms of, if you were analyzing what is the threat to honeybees foraging in these areas. A surprising result, an important result. I'll talk more about it in a minute. Now, what plant species were we looking at? One of the things we wanted to do with this work is map the plant species along with the pesticides that we found. So we wanted to see, for example, if one of the bees are foraging on corn, do they get a spike in neonicotinoids? When they forage on certain weeds, do we see other compounds coming in, herbicides perhaps? Now, in the non-agricultural area, you can see it's dominated by wild mustards, brassicas of various types that they forage upon. Plantain, shown in the sort of blue-green on the 9th, 16th, and 23rd of August, is also a big player, and that'll come in again as well. So the purpose of this work is also to show that honeybees are extraordinarily resourceful at finding a wide range of plant species. If we all went out and looked for these species of plants in, let's say, a 1.5 kilometer area, I think we'd have a really hard time. It was really amazing to see the diversity of sources that these bees used. The untreated cornfield, you can see different plants are used here. The clovers are a big player here. You can see the light purple and the bright yellow are both clover species. You can see corn shows up here as well. That's the sort of neon green with the black asterisks embedded in it. We did not see a strong synchrony between crop pollens, corn and soybean, and agricultural pesticides. So what I think that reflects is that the agricultural pesticides, I think largely in the case of neonicotinoids because of their water solubility, are expressed in other pollen types as well, and there's not that synchrony. They're not only in corn and soybeans, in other words, so you do see them popping up at other times in the season, and I think that's because of water movement and uptake by the weeds. Here's the treated cornfield. Again, we have similar clovers and agricultural weeds are dominant, and plantain is a big player here. Again, 2nd, 9th, and 16th of August, you can see large amounts of plantain. I was struck by how much the bees really gravitate toward that plant. It's a real key pollen source for them when it's available. Okay, now I've thrown a lot of data out, so I'd like to summarize some of it, hopefully tie some of this together and get into the what do we do next, what do we do with this information type of message. So the first set of research that I talked to you about was planter dust, and we've known that was a hazard for a while. There have been some efforts to work with other non-talc and graphite products to accomplish the flowability of seed. Those have met with limited success, limited adoption. So talc and graphite, we're still planting seeds on the overwhelming majority of acres the same as we did in 2012. This exhaust moves neonicotinoids well beyond planted fields, so safe zones are not a realistic goal. In Indiana, and I would suspect in other similar states with similar agricultural constraints of a lot of corn and soybeans, there are many intersections between honeybees, foraging, and lethal doses of these insecticides. The use of new lubricants and or polymers would reduce drift, as I said, but adoption is relatively limited right now. One solution that is very apparent from this work and other work is that aligning the use rates, which are currently near 100% in corn, with target pest incidence is the most efficient approach, and that not only from the pollinator perspective and from the perspective of contamination of water and so forth, but also financially, as commodity prices are lower than they've been in the recent past, and this represents a financial outlay that it doesn't seem that many growers are benefiting from at present. In terms of in-plant concentrations, the window is shorter than perhaps many would have anticipated for concentrations of the seed treatments in plants. And this, coupled with the scarcity of target pests, I think explains why we can't find the yield benefits or why they're so inconsistent, because you have to have a very short-lived pesticide in the plant and you have to have relatively uncommon pests. Those two things have to overlap. Those two circles have to happen. And have to happen to overlap, and that rarely happens in most of the, certainly in our studies in Indiana, and anecdotally in other areas of the Midwest as well. There are some pest concerns, some pests that are rising in prevalence in corn and in soybeans. Slugs are notable there, and John Toker has a NRCS webinar that will talk a little bit about those. They're not insects. They're not managed effectively by neonicotinoids, and what Dr. Toker has is some evidence that show that they benefit from the use of neonicotinoids because the neonicotinoids wipe out their predators, carabid beetles. We have Bt resistance in corn, rootworms, and ear-feeding caterpillars. There's no role for the neonicotinoid insecticides in mitigating those in populations of those insects. So we don't really see a fit there either. Treating the seed seems to be a more inefficient way to deliver these compounds than initially thought. And this is primarily because of water solubility. Although water solubility can be used to get the material up and into the plant, the problem is when a plant is saturated at any point in time and you get rain or water movement of any kind, that material is going to move away from the plant. And you can see I've got the water solubility values here. You can see it's very high for thiamethoxam in particular. So these compounds will go into solution. This is just the physics of the compound and can't be dictated by anything other than a saturated plant. These compounds will then go to an area of lower concentration and move out with water and into areas where the concentration is lower. This is just simple physics. Our pollen sampling told us that, number one, there's a wide range of pollen types gathered by honeybees, even when we put them right beside cornfields. Now, this was all done after the period of dust and planting was over. We only started this study in late May of that year, deliberately to get past that window. We wanted to see what happens after that. We saw contamination of the pollen with multiple pesticides. Municipal and homeowner pesticides were at the highest concentrations and posed the highest risk to bees. We did find clothianidin had posed a high risk at those sites, but the big player here, and surprisingly, were these municipal and homeowner pesticides, reflecting that any changes that would happen in terms of pollinator health and non-target organisms and so on at the ag level would have to be mirrored at the level of municipal and homeowner use, because it seems that this is a much larger contributor than perhaps some of us would think. Interestingly, and again going toward that same theme, the mosquito repellent DEET was found at every sample. This indicates that honeybees go where people are. Our hypothesis is that they're picking up DEET at areas where humans are bathing and water sources that they're using. DEET wasn't acutely toxic. It was just a marker that bees are going where people are and where people have been in the water. We found strobilurin fungicides in every sample as well. These, again, aren't acutely toxic, but they do have some synergy with some pesticides, including neonicotinoids. So, again, reflecting that looking at pesticides one at a time is not likely to provide a complete answer in the case of pollinator toxicity. We had no instances without multiple pesticides per sample, and so, again, synergy is likely to be very important here. We don't have a lot of opportunity to improve currently because the rates are so high and these acreages of these crops are so large that there's not much we can do without at least addressing that disconnect between use rate and need or benefit or pest incidence, however you want to put it. Making this untreated seed widely available would offer an opportunity for correction. I believe that many producers would find out very quickly whether they benefit from using these products. A simple side-by-side comparison in their fields would quickly reveal what benefits are there, if any. There are very few of those studies that exist, and even fewer that are published. And that's something that we can easily address, and we can get some answers for everyone, all the stakeholders involved in the use of these products. However, the problems of these compounds of high water solubility and contamination of water and very high toxicity of pollinators means that they have these troublesome traits. That means that the margin for error is small, and there's no getting around those facts. There are things that can be done. They start with reducing the use rate, but an acknowledgement that we have these particular traits of these compounds, I think, is a first step in trying to develop some ways to use them more efficiently, more judiciously, and in a way that's smarter, to put it bluntly. In terms of regulation, this seems to be where a lot of the questions come. Are there regulations coming? Will regulations change the way we use neonicotinoids? Well, I'm going to start by looking at other regions in the EU. There's a moratorium on many of their annual crops in 2015 and 16. That's currently under review. I believe I don't have the latest update on that. In Ontario, Canada, there's a requirement now for growers to scout the fields in the year prior to buying seed and confirm pest presence. This is thought of as a prescription for using neonicotinoid-treated seeds. So pests are documented. If the target pests, and these are things like wireworms and white grubs, if they're documented in the field, the grower can use that information, that monitoring information, and purchase his or her untreated or treated seed. Minnesota is the first state to address this. The governor wrote an executive order last year that may, I'm guessing, result in similar restrictions as Ontario. I don't know what way that is going, but the executive order was simply to limit neonicotinoid use. It didn't contain specifics on how that should be accomplished. Finally, federally, there's a review at the EPA continuing of the conditional registration for clothianidin, which was granted in 2003, but this re-review goes on. The next update is expected in 2018. So I don't have anything further to share with you other than they've been working on it for quite some time, and periodically we get updates reflecting the new information and some new guidance and new testing, particularly aimed at pollinators. All right, that's all I have. Thank you for your attention.
Thank you, Christian. Excellent presentation. We did get some questions. The first is, and I think you addressed this a little bit in your presentation, but I'm wondering, what is the minimum insecticide-free buffer needed to protect sensitive areas such as pollinator habitat?
Right. I tried to get at that a little bit. The minimum insecticide-free buffer, if insecticide-free is your bar, I'm afraid there isn't a number that I can give. Not to be overly simplistic or trite, but farther away is certainly always better. Any kind of buffers in terms of hedgerows or tree lines are beneficial when we talk about the dust, but because of the use rates and because of the acreages that we're talking about, an insecticide-free zone in terms of neonicotinoids and pollinators is not really a realistic goal at this time.
Okay, thank you. We have a couple questions related to the generality of your findings. And are your findings relevant to other parts of the Midwest and other parts of the U.S.?
I think to the extent that much of the upper Midwest and stretching over into the Great Plains are corn and soybean states, quote unquote, I think they're generalizable there. Indiana is in the top 5 in both corn and soybeans, and so I think it compares well with many other states in that respect. I think our pollen data probably, for example, this pollen species would change a bit. But I think the fate of the compound on the seeds, the movement in planter dust, I wouldn't expect those data to be very different as we move from state to state. There are some regional differences in pest abundances, but a lot of the data are, I think, generalizable and certainly can at a minimum be used for informing future work in those other areas.
Okay, thank you. One of our participants asked, have you done any work assessing risks to native bees?
We have not. We're just starting to assess what native bees we have, and we're doing that in soybeans because we see, of course, soybeans have flowers. They produce a little bit of pollen and nectar. It evidently is enough to interest some of these small native, mostly solitary pollinators. So we're beginning to look at what those species are. We do know that these insects are much smaller, generally less tolerant of insecticides and larger, more robust bees like honeybees and bumblebees. And they're generally solitary, so they don't have that colony of 40, 50, 60,000 to buffer toxicity. So we might, and finally they live in the ground, many of them do. So we might expect them to be impacted more based on those features, but impacted more by neonicotinoids, but we don't have good data on them. We don't have good life history data, and we're just starting to untangle their intersections with these crops.
Thank you. How are seed treatments applied, and are methods of application the same for corn and soybeans?
They're very similar. They're applied primarily now and increasingly. It's a centralized operation where it's done prior to sale to growers. It's not done by private growers. It's done by specialized, large, sophisticated equipment where they can handle large volumes of corn and soybean seed. So in the past, I believe, let's say 10 years ago, there was some on-site or smaller scale treatment, but now it's, I believe, largely centralized in just a few locations per manufacturer.
These neonicotinoids are persistent in the soils and apparently accumulate. And can you comment on the effect on invertebrate and microbial community in the soil?
Yes, that's an area that, again, is just starting to get attention. We know earthworms, for example, are sensitive to neonicotinoids. Some other soil organisms, collembolins, and some of the other decomposing organisms haven't really been examined closely to see what are the effects of a soil, a relatively persistent, relatively widespread soil insecticide. Again, similar to the question, or similar to my comment earlier that we don't have pest data going back to 2003 on these soil pests, we don't have it for the beneficial organisms either. So there may be changes that have happened there. They may be positive, negative, or neutral, but we just don't have those longitudinal data. But we're starting to see some manipulative experiments, and I think in the next 2 to 4 years you'll see some publications that reflect whether these compounds do affect these soil organisms. You would think there would be some effects given that they're living there, and this is a relatively new class of insecticides that's gone from zero to ubiquitous in about a decade.
Yes. You mentioned this during your webinar, but I'm wondering if you can repeat, some of your guidance regarding steps that can be taken to minimize aerial drift of pesticides onto adjacent areas.
Right. So you're speaking in terms of the planter dust component?
Yes.
Right. Wind, velocity and speed is always a factor that's unpredictable. We did see a trend with more wind velocity. We got more deposition. That's not really news. Anytime that you can have interference, physical barriers, hedgerows, and trees and so on between an area being planted and sensitive organisms like honeybees, that's beneficial. Some of the alternative lubricants, fluency agents and so on, reduce the amount of dust drift as well. More adoption of those would help. But again, the glaring and striking example of what we can do is the reduction in use, because we have such a hard time quantifying the yield benefits, and yet they're used on virtually every corn seed in the country. And so that disconnect it could be remedied, and then all the things that I just talked about, those other methods, don't even need to be addressed because you don't have any insecticide to drift, and you've gone from some risk, some level of exposure to zero.
Yeah, thank you. Here's a question related to the risks of the seed treatments to aquatic organisms, and during your webinar you referred the listeners to Dr. Morrissey's companion webinar that's also on the conservationwebinar.net site. So I'll pass on that. Can you comment on the relative risks posed by surface versus subsurface runoff?
We haven't done those actual comparisons. I can't really go into too much detail on that, like in terms of what component is surface runoff versus subsurface. We can say that both are factors and that both are potential issues. But in terms of quantifying the levels and saying, that one is on this order of magnitude versus one on another order of magnitude, we don't have that. We do have data that show that that indicate a lot more subsurface movement than we thought, because we pick these compounds up far from agricultural sites, which indicates they're moving in subsurface, and then we're finding them later in areas of drainage. So they are relatively stable if they're not exposed to UV light, and so if they move below the surface, those compounds will remain in solution in water and will be stable until they are exposed to light.
Finally, Christian, do you have a sense for what these seed treatments cost producers?
Yeah, that's a good question. And because in corn the cost is never split out because it's on all corn seed, it's difficult to assess, and we don't have a good hard number there. It's like asking, how much does the salt on my fries at McDonald's cost me? You don't have it with a version without, so you can't really get that number. We know that in soybeans, it can range from the teens per acre, the high teens dollars per acre upward, and in corn, we don't have those data to compare. We know that it obviously costs something, but we don't have good data to share on that.
Well, I think we'll leave it there, Christian. I want to thank you for an excellent, informative webinar, and I greatly appreciate your sharing your cutting-edge information with us.
Thank you.
You provided your contact information, so with your permission, I'll encourage our listeners to contact you directly with their questions,
yes,
should they have it.
Please do so. Yeah.
Great. Thank you again.
Thank you.
Okay, thank you, Christian, for making the presentation today. It was really very interesting. And Bill, thanks for coordinating another very interesting and successful webinar. Thanks to all of our participants for joining in. We had more than 450 people join today's webinar. Participants, provide your feedback about the webinar, and if you selected to earn CEUs, please return to your open browser window to continue the process that's offered by Step 2 at conservationwebinars.com.